the second class of my and I hope you yeah I hope you you came to understand that there's a huge variety of plant pathogens, and therefore the plant immunity is highly complicated and sophisticated. But today, what I would try to do is to further increase the complexity by introducing other microbes, like non-pathogenic microbes that also interact with the plants, and that include symbiosis, plant microbial symbiosis, and the plant microbiota. And I will try to explain everything that I personally find interesting and some of the recent works that we have been doing in our lab. But again, if you have any questions or comments, if you want to discuss anything about the content of this research or my research, please feel free to contact me via any ways you want to use. Good. So shall we start with this plant symbiosis? So plants, the symbiosis in general, particularly, or if you say symbiosis, that implies that it's a mutualistic symbiosis, which basically means that there are at least two or more organisms that provide benefits to each other. So both parts of these interactions take benefit from the interactions. And that's the definition of mutualism. And if you say symbiosis, you already imply that it's a mutualism. And there are actually many examples of such mutualistic symbiosis between organisms. For example, between animals and fungi and bacteria, animal and bacteria, and plants and animals. But of course, today, what we want to focus is the plant microbe symbiosis. Particularly, we're going to focus on Rhizobia nodule symbiosis and the avascular mycorrhiza symbiosis. So avascular mycorrhiza symbiosis, or AMF in short, is a very common and widely distributed symbiosis. So, for example, here these are maize plants. On your left-hand side, they are the plants that are associated with or establishing symbiosis with avascular mycorrhiza, while on the other hand side, on the right-hand side, are the plants that do not engage in avascular mycorrhiza symbiosis. And you can easily recognize that the plants with symbiosis are more greener and more like healthier, while without symbiosis, they show pale green, a little bit sick phenotypes. And if you focus on the yield, these maize plants produce beautiful corns, while on the symbiosis-less plants, they provide small, sick corns. So what these mycorrhizal fungi do is actually to provide the plants with the nutrients that these microbial hyphae or mycelium collect from the soil, right? So on one hand side, the fungal hyphae is in the plant cells, while the other hand side is distributed or spread into the soil. So they take up the nutrient from these hyphae and transport all over this hyphae, and then provide them to the plant host. And in most cases, these are phosphates that are mostly important for avascular mycorrhizal symbiosis. For example, if you put a huge amount of phosphate to the soil, then this avascular mycorrhizal symbiosis is basically gone. So phosphate is one of the key factors for the symbiosis. Plants, on the other hand side, they provide carbons or organic acids or lipids that the avascular mycorrhizal fungi cannot synthesize on its own. So again, this is the mutual symbiosis between plants and avascular mycorrhizal fungi. So what's interesting about this AMF is the fact that more than 80% of land plants are engaged in avascular mycorrhizal symbiosis. Sorry, there's a typo here. So if you simply take some roots on the natural side and stain avascular mycorrhiza fungi with a blue dye, then you can appreciate that many of those root cells are filled with fungal hyphae, like these structures, the structures called avascules. Oh, and by the way, these photos are borrowed from the Twitter account of Takeshi Sentoku. He always posts beautiful mycorrhizal fungi pictures, and I love it. So, great. And so, like 80% of land plants are engaged. So it's really common and widely distributed throughout the plant kingdom. And what's also fascinating is that there was a people like almost 30 years ago looked into the fossil record. So they took one of the first generations of the land plants, like it's 350 to 450 million years ago, and then looked into the cells, the root cells or root-like cells of those fossils, and found that there was a fungal structure that is very similar to the avascules that we found in the extant plant roots. So this indicates that the symbiotic interactions or mutualistic interactions between plants and avascular mycorrhiza was already established right after the plants start growing on land, or plant terrestrialization. Excuse me. And that's one of the reasons why most of the land plants are engaged in mycorrhizal symbiosis. Good. So that's one part. So it's very common symbiosis. And the second example that I want to introduce is rather more specific symbiosis, and it is specific to legume plants. So legume plants is the like beans, so to speak, soybean, mungo beans, medicago, alfalfa, so on and so forth, you name it, and also clover. So if you take out a clover, for example, from your garden or from your street or from the park, and carefully look at the roots, then you will find these globular structures on the roots. And this structure is what is called the nodule. And if you cut the nodule and look inside, then you can find this pink area. And these are the area where bacterial species called rhizobia are accumulated, right? And they are accumulated in the plants. I don't want to go to too much in detail of the symbiosis, but basically this structure is filled with bacterial cells. And what these bacterial cells do in the plant cells is to fix atmospheric nitrogen gas. Oh, sorry, right? So you have a nitrogen gas, and then this bacteria shown in orange, they transform the gas into ammonium, and then this ammonium is transported to the plant cells, and then this is utilized by the plant as a nitrogen source. And there's only limited organisms that can do this nitrogen fixation. And if you don't have enough nitrogen in the soil, then of course plants will suffer from nitrogen starvation. But if you have this rhizobia on their nodules, then they can take nitrogen not from the soil but from the air, from the atmosphere, and then survive in the nitrogen starving conditions. So this is how it is. Then of course again plants provide carbon sources, the photosynthate, in the form of organic acid, and then this rhizobial bacteria cell utilized this organic acid for their own energy. So these are like two extreme examples of plant symbiosis. Both rely on each other, providing essential nutrients for their growth and for their survival. If you are interested in symbiosis, check out some textbooks or review papers. But the question here is, like, if you think about what we have already discussed in the first class, where a plant has an immunity to recognize microbes and to respond to microbes and to attack the microbes to restrict their colonization or invasion or infection. So the question here is how, or whether and how, plant immunity recognizes these symbiotic interactions and whether they do something on it. And the answer is yes. So here is a schematic representation. Here's the plant cell and the rhizobia. And what actually happens is that the pattern recognition receptors at the plasma membrane, you remember the surveillance system at the plasma membrane looking outside of the plant cells to be ready for recognizing the microbial pattern, this molecular pattern. And this rhizobia is indeed recognized by pattern recognition receptors, and they trigger defense reactions like PTI, the pattern triggered immunity. And the rhizobia typically have type 3 or type 4 secretion systems to inject the effector proteins into the plant cell, just like the pathogens do, right? They are mainly not effectors. And these effectors are able to compromise the PTI responses. And again, like an ETI against pathogens, the legume plants also do have intracellular receptor proteins called NNR and trigger ETI, the effector triggered immunity. Right, so this is like identical to what we discussed in the first class, right? You have microbes and receptors that can recognize the presence of microbes. They trigger immune responses, and rhizobia inject effectors to manipulate these pathways, and the plants recognize these effectors to trigger further immune responses. So it's exactly the same thing is happening. Now the question is, what was the consequence of such activation of PTI or ETI? And on the right-hand side, what you can see here is soybean plants are inoculated with a rhizobium strain called USDA 122. And it's been known that this USDA 122 cannot establish symbiosis with this particular cultivar of soybean, hardy RJ2. And well, of course, they didn't know the reasons. But here, what was reported here is that if you knock out the effectors or the secretion system, so these two strains of bacteria are unable to inject this particular effectors to the plant cells. And the consequence is the reactivation of the symbiosis, right? If you have the effectors that trigger immunity, ETI, you have no symbiosis. If you don't have the effectors, if you don't activate ETI, then you can engage in symbiosis. So if you have ETI overactivated, then you don't have symbiosis, which basically means that the plant immunity needs to be properly regulated to maximize or to obtain to achieve the symbiosis. So that's what we know about the role of plant immunity in plant symbiosis. Of course, there are many unanswered questions, like why in the first place do plants recognize the effectors and trigger ETI? Because the activation of ETI has a fitness penalty to the plants, right? They want to have the symbiosis, they want nitrogen, but they activate ETI to restrict the symbiosis losing the opportunity to get more nitrogen. There are a couple of hypotheses, but at least for me, none of those hypotheses has convinced me just yet. So I'm very much looking forward to the further development of this research field. But the take home is the plant immunity is activated not only by pathogens but also by symbionts, and the activation of plant immunity has a consequence on symbiosis, just like they have a consequence on the plant virulence. Okay, now then here I would like to further complicate things. Sorry for that, but I'm trying to do this because I believe this is very important. And so if you think about the natural condition, like this is Arabidopsis thaliana growing outside, then if you take out the soil and see what's there, there are huge dose of microbes, a huge variety of microbes, a huge abundance of microbes, including bacteria, fungi, oomycetes, nematodes, algae, cyanobacteria, you name it. And this is the environment where plants develop their roots. And indeed, if you take out the roots and stain microbes with a fluorescent dye, and then you can see that roots are heavily colonized, literally heavily colonized by many soil-borne microorganisms. And this is the condition where plants are, ensuring their survival and growth. So what are these microorganisms? There have been many studies showing that they indeed include some beneficial microorganisms, for example, like plant growth promoting rhizobacteria or PGPR. And also there are some opportunistic pathogens in the system. I will come back to this point later, but there are some microbes that can be pathogenic under certain conditions, but not always, because these are the healthy plants, right? But the majority of these root-associated microorganisms is somewhere in between. They are not explicitly pathogenic, they are not explicitly beneficial, but they are still associated with plant hosts, and they establish their niche. They win the competitions against other soluble microorganisms. But whether they do provide any benefit to the plants or not, that's a big question. That's a question mark. I mean, I'm not doubting, but we just don't know what these bacteria or these microbes are doing, and that's why we call them as a commensals, meaning that plants provide benefits to bacteria, to microbes, but it remains unclear whether microbes provide benefit to the plant. So it's just one direction of benefit. That's why we call it as commensals. And the fact is that plants are interacting with a community of these microorganisms. So different lifestyles, different taxonomy, different growth rate, different functions. It's a mixture of all these microbes that are interacting with the plant host under natural conditions. And that's the interaction that I personally want to understand. So why this is so important? And I will give you a few examples of how these microbes can affect plant physiology. This is my previous study where I inoculated Arabidopsis plants with a variety of root-associated commensal bacteria and see what happens. So I individually inoculated. So to make a long story short, what we found is that the root growth is strongly promoted by these commensal bacteria, a group of commensal bacteria. So this is axenic plants without any microbes, but in the presence of microbes, this root are much longer than these axenic roots. This is a weird direction of the roots. This is just because I had to move these roots to take a picture. So it was actually quite straight on that. But anyways, the primary root growth is strongly promoted by Rhizobialis commensal bacteria. Whether this is benefit to plants or not, that's a big that's a question, and we don't know. At least the plant root growth is promoted, but not the shoot growth, for example. So who knows? But this is the activity that root associated commensal have. Another example, maybe we can start from the right hand side. These are again Arabidopsis plants. And these two pictures, these are normal media, and the microbes do not have a strong impact on plant growth. They do have a bit of difference in the root architecture, but more or less the same in terms of growth. On the right-hand side, this is iron starvation conditions. So the iron is there, but plant cannot utilize those iron in the media. And therefore, plants are strongly suffering from this iron starvation. But if you add bacteria into this media, then this bacteria help plants solubilize their iron in the system and thereby rescue the plant growth, even under these harsh iron starvation conditions. So there's a strong and clear support for the plant growth under these particular conditions. On the left-hand side, this is another example. So here, again, on two pictures on the left are without any microbes, and two on the right are inoculated plants. And here what we try to do is to chronically activate it or continuously activate the plant immunity. And the consequence of this is growth inhibition. Right? I will come back to this on the next slide, but there is a growth inhibition when the immune response is overly activated. This has been known for like quarter of centuries or a couple of decades. Okay, but when we did the same experiments in the presence of those root-associated commensal bacteria, this growth inhibition is simply gone. Right, even in the presence of this immune activation conditions, the plant growth is not inhibited, and they can ensure the growth as good as a normal control. So as you can see from these examples and the previous examples, these root-associated commensal bacteria has a strong influence on the plant growth and plant physiology, right? So coming back to this immune activation, so why do plants show this growth inhibition and the chronic activation of plants? And that's believed that is believed to be a consequence of so-called the growth defense trade-off. So it is really a conceptual idea that plants need to prioritize either growth or defense depending on the surrounding conditions. So if they decide to defend, then they need to prioritize their resources to defense, and therefore they have to restrict or slightly downregulate the growth. And that is why you see a growth inhibition when the immunity is chronically activated. So like one of the questions that I had was, when these bacteria can inhibit this growth inhibition by immune activation, it could simply be like suppressing defense, and thereby shifting this trade-off not from defense to rather to the growth, right? Just simply suppressing defense to achieve growth. But this seems to be not the case. So this is a little bit complicated data, so it's a bit difficult. But here what we try to do is to evaluate the immune activity by looking into this gene expression. So if you see blue colors, it means the immunity is activated. If there's no blue colors, then it means there's no immune response is activated. And you always have oxenic control and inoculated plants, normal condition or immune activated conditions, right? You don't have to understand everything. All I want to say here is that even in the presence of this bacteria that can suppress the growth inhibition and rescue the plant growth under chronic activation of plant immunity, you still see a strong activation of immunity. For example, here and here, you see blue colors even in the presence of this bacteria, meaning that it does not suppress immune responses, but it does suppress this growth inhibition triggered by immune responses, probably by interfering with this growth defense trade-off and reallocating on the resource or the coordination of different things. We don't know how, and that's exactly one of the projects that we are running. We have been running for 5-10 years for now in our group, and I hope to provide some evidence in a couple of years. Anyways, so this is excuse me. So from this data, I hope I could convince you that this root-associated microbial community does have an impact, or is important for plants, for plant growth, for plant immunity, for plant physiology. And again, the question is how plants deal with them. Like, how do plants recognize these microbes? Do plants induce immunity? And so on. So there are a couple of studies that try to address that question. And this is from Sebastian from, at that time, Zurich, but now in Amsterdam. So he took a mutant impaired in immunity. This is RBOHD mutant, which is impaired in production of reactive oxygen species. You might remember this immediate responses, and grow them in oxenic condition in absence of microbes or in the presence of microbes. The microbes that are isolated from healthy plants, the commensal bacteria. And then you see wild-type plants are totally healthy and fine, but the mutant showed yellowish disease symptoms, or sometimes very small sick plants. This is also the case for the pattern recognition receptor mutants, where plants are not able to, or plants are incapable of recognizing those molecular patterns from the microbes. And when they inoculate with the commensal bacteria community, again, Columbia plants, the wild-type plants are fine, but the mutants show very diseased symptoms, indicating that, okay, immunocompromised mutants show impaired growth, and therefore we can say that plant immunity does play a role in interaction with plant commensal bacteria. So, but then the question is, because the concept is quite different. Plants need to restrict pathogenic invasion by activating plant immunity, but at the same time, plants actually want to accommodate those symbiont or beneficial microbes, and also those root microbial community. They need to manage the microbial community by plant immunity. So one of the biggest questions that we had, so this is actually an international congress on molecular plant microbe interactions held in 2019 in Glasgow. So these are my colleagues who participated in the meeting. And there we did a kind of interesting experiment where we come up with the top unanswered questions in molecular plant microbe interactions, and we voted what would be the top unanswered questions. And the way top than all the others was this question number 1, which goes, how do plants engage with beneficial microorganisms while at the same time restricting pathogens? So how plants can discriminate pathogen versus non-pathogens, and including microbiota members. And that's like $100 million question in the research field, and we still don't understand it. But this is one of the biggest questions. And me personally is also so my interest personally is how these microorganisms, or how do plants regulate their growth and defense in the presence of a complex microbial community? Because if you have this immune activation, you might have growth inhibition, while the plant immunity might be actually continuously activated by root commensal bacteria. So how do plants coordinate this? And in other words, if I put these into question in the form of questions, the first question is how do plants and microbiota communicate with each other? And secondly, how do plants coordinate growth and defense in the presence of microbiota? And the third question is, because it's a microbial community, like I said in the beginning, at the beginning, it's a mixture of microbes with different lifestyle, different functions. So how are individual microbial traits or functions to manipulate host plants regulated in the presence of other microbes? The community context function expression, that's one of the key aspects of this microbial community. But as I said, this is a microbial community. It's really a complex microbial community, and it's very technically challenging to work with the soil because of the reproducibility issues and many other issues. It's been very difficult and challenging. So the approach we employ, and my former lab in Max Planck has developed, is the culture collection. So we basically grow plants in natural soil, homogenize leaves and roots, and try to collect microbes that colonized the leaves and roots of Arabidopsis. And then we purified them, and we made them into a culture collection. So these are just a couple of examples, like different bacterial cells individually cultured. There's Actinobacteria, Pseudomonas, Xanthomonas, Sphingommonas, and Rhizobium, and this is Actinobacteria. We can grow in liquid or on the plate. Oh, there's a contamination here. Anyways. And we can also, because it's a purified culture, we can do genome sequencing. We have 200 root isolates and 200 leaf isolates from Arabidopsis, 1,000 Rhizobiales isolates from many different plant species. You have 200 species from Lotus japonicus, 200 species strains from Chlamydomonas, and many others. So we have now like thousands or dozens of thousands of microbial isolates whose genome sequences have been completely sequenced, and we have all the cultures available. But then what do we do? How do we use this culture collection to understand plant microbiota interactions? So the best advantage of this culture collection is that we can actually make a synthetic community of microbes, which basically means that we grow individual microbes separately and then mix them in a way that we want. Like we can choose like 3 of them, mix them at equal ratio, or take 20 of them and mix them in whatever ratio you want. You can have like 90% of one isolate and the rest was dominated by other microbes, for example, right? And then we inoculate plants with this microbial community, the synthetic community or SYNCOM, in an axenic or gnotobiotic system. So like autoclaved soil, autoclaved clay, or the agar plates that we normally use for cultivating plants. And I believe this is a really, really cool system where we can reconstruct the plant microbiota interactions in whatever conditions that we want. We can introduce nitrogen starvation stress, iron starvation response, chronic activation of immunity, highlight stress, drought stress, salt stress. We can also put immunocompromised mutant, mutants impaired in whatever secondary metabolites or cell wall or autophagy, whatever. So everything is possible. So using this system, and what is most important is that we can do this in a reproducible manner, right? We culture them in a fixed ratio, make them SYNCOM in exactly the same manner, and grow them in a controlled condition, which is a huge benefit compared to working with the soil or working in the field, right? So nowadays we have culture collections and you have a system for reconstructions. So we now can ask whatever questions we have. And therefore, what is now the most important thing for now is what kind of question you want to ask. So whether you provide a meaningful results or not depends on whether you have a meaningful question or not. And that's, of course, challenging, but it's also, the major reason why this research field is so interesting and fascinating. Of course, I have a handful of different questions, but I can't tackle all of them by myself. So I'll be happy to have more members and more colleagues to tackle as many questions as possible. So yeah, that is the current status of the research area. And at the end of this talk, I would like to share my thoughts about the possibility of translating this research into applications, for example, to agriculture, right? Plant microbial instructions is always associated with agricultural practices. And, there are many microbial community that provides a penalty to plants, what we call as bad community, where you have low yield plants or high disease frequency. And you want to change this. You want to make them into good community, or you want to improve the yield or disease resistance. And of course, classic or canonical method is to use pesticides, fertilizers, like chemical fertilizers, and those things. When it's expensive, it's energy not energy friendly and not environment friendly. So the idea was to use those root-associated microbes with some beneficial functions to transform a bad community into a good community with high yield and low disease frequency. And that's been the idea. And there has been many products actually, and that to that end, using these microbes for the agriculture, and that's called the biofertilizers. However, those products, like biofertilizer products, is not widely used by the farmers. And one of the reasons for that is the biofertilizers provide, not as robust and reproducible impact on crop growth as we hoped. Like sometimes it works, sometimes it doesn't, and you can't predict whether it works or not until you actually try this. And if you are a farmer, would you like to use an expensive biofertilizers that you don't know whether it works or not? And that's why farmers still use chemical fertilizers a lot. They are not keen on using biofertilizers that much. And that's why biofertilizers are still expensive, and R&D is not, progressing as we expected. And of course, there are question of why this biofertilizer is not reproducible, and one of the reasons is the presence of soil indigenous microbial community, right? So we'll just start this. Sorry. So the idea is probably I think there are two reasons. One is that the biofertilizers cannot invade into the community, and establish their position in the community. They are basically kicked out from the already established community. Another scenario is that these biofertilizers can actually invade and establish their position and persist in the community, but they cannot express its function, right? And so these are two possible scenarios. And so we did some experiments in this direction. So we started with this fact. So as before, we have a lot of microbial isolates derived from healthy Rhabdopsis thaliana, and we inoculate these bacteria with arsenic Rhabdopsis in the chronobiotic conditions, and they found that actually quite some of those root-derived commensal bacteria are pretty pathogenic when they are inoculated alone. This is Pseudomonas, this is another Pseudomonas, and this is Xanthomonas, all derived from healthy plants. Sorry. Indicating that, or suggesting that the pathogenic activity or pathogenicity of these bacteria are suppressed under natural conditions in the presence of other microbes. And this is the example of bacteria incapable of expressing its functions in the presence of other microbes, right? So we are currently working on why this is the case, how this is regulated. So for that, for a couple of reasons, we are using duckweed plants, with aquatic plants floating on the surface of the pond or river. You can also find in Hokulai campus or anywhere in Japan or around the world. So we inoculated this duckweed with root 401, which is an opportunistic pathogen. You can see the duckweeds are brownish and very diseased. And in the presence of Syncom, this is a duckweed commensal bacterial community, plants are fine. And if you mix them together, then still plants are fine, even in the presence of root 401 pathogen. So this pathogenicity of root 401 that is expressed when it's alone is strongly suppressed by co-inoculation with commensal bacterial community. We are working on why, and hopefully we will be able to answer this at some point. But this is, again, a clear example of functional expression of the community is dependent on the other microbes in the system. We also have this experiment. This is the phenotype that I already explained to you, where plants growth is inhibited by chronic activation of immunity in the absence of microbes. But in the presence of one particular bacteria, then this growth inhibition is not present, right? This is a growth inhibition arsenic condition. There's no growth inhibition. Okay, so this is a suppressive activity. We got two more strains where you still see this growth inhibition. So they do not have this activity. But only this purple one can inhibit this growth inhibition. Okay, and in this experiment, we mix this purple one and the blue one. So this the one with the activity and the one without the activity, and the consequence was the suppression of the growth inhibition, right? Like this magenta one, you don't see the growth inhibition anymore, which makes total sense, right? You have, if you have plus and minus mixed together, then that's a plus. But interestingly, when we did the same experiment using the sky blue isolate 71, then this growth inhibition is suddenly gone, even in the presence of this 1497. In other words, the suppressive activity of 1497, the ability to suppress this growth inhibition by immunity, is inhibited by co-inoculated root 71 isolate. And this is, I think this is very fascinating, that whether this 1497 can express this function or not fully depends on with whom they were co-inoculated together. So depending on who's around, your behavior will be totally different. So this is also an example of the functional context, community context function expression. And one of the major goals that we have now is to understand what dictates this difference, these two differences. What is actually different between these two conditions, and why 1497 can do the job with 142 but cannot with 71. Okay, good. Now, so that's the idea. To achieve more effective biofertilizers, or to better utilize these microbes for agriculture, we need to understand how microbes can invade into the community and how they can express their functions. And by understanding the molecular principle behind, then we can probably manipulate the community such that the biofertilizers are easier to get into, right? Not make the closed community into an open community, so biofertilizers can actually go into the community and stabilize. And also, after they invade into the community, they still need to express their functions. And my idea is that there must be some bacterial combinations that support the functional expression of each other. So not like applying not the single isolate, but rather we can apply a combination of microbes so that so one bacteria can support the functional expression of the others, right? And I'm not even sure whether we can actually do this, but basic idea is something like this. And this is the idea of how our research may possibly contribute to the society. And you might have actually realized that I'm using this metaphor of human, right? But of course, it's not good to do that because bacteria are not human. Bacteria do not have brains. Bacteria do not really intentionally regulate or try to maximize whatever. But I'm actually intentionally doing this because what's been discussed here is very similar to sociology, right? Sociology or social science is the science that tries to understand the behavior of society or the community by understanding the individuals. Right? And there's a mathematical sociology where you model the community behavior based on the individual behavior. And not only the individuals, but the interaction between individuals, like one by one or one to one or one to several. And this is exactly what we are trying to do, right? We want to understand the microbial community behavior by understanding the individual microbes and the interaction between microbes. And there are theoretical framework in this mathematical science. And one of the idea is to utilize those framework to explain the microbial community behavior. We just started this project very recently, and I'm not sure if it works or not, but hopefully at some point we may be able to find something in this direction. So anyways, that was it. I hope I could convince you that in the beginning I explained there are interactions between plant and microbes, not pathogenic interactions, but there are symbiotic interactions and the commensal interaction with the microbial community. And many of the data demonstrated that the plant immunity is involved in the interactions not only with the pathogen, but also with symbionts and microbiota. Just skip the second point. And also we showed that this root-associated commensal bacteria can also not only immunity regulate microbes, but the microbes regulate the plant growth and plant immunity. So there's a the bidirectional communication between them. And that's the molecular mechanisms behind or underlying this communication or mutual regulation is what we really want to understand. And thereby addressing the role of root immunity in plant microbiota interactions. Oh my God, so many typos in this slide. Sorry for that. Yeah. So that's it. That's the current situation of the research field. So this plant microbiota research just started in 2009, 2010-ish. In the current approach, of course, it's been a long history of century of history of plant microbiota interactions, but over the last couple of decades, the techniques and ideas have been developing like crazy. Some may call it skyrocketing, and things that were impossible 20 years ago, it's super easy now because of the culture collection, because of the sequencing technology, because of the many other things. So there are many things that we still do not understand. And I believe that the concept of plant immunity is quite different between leaves and roots. And the role of immunity in the pathogenic interactions versus microbiota interactions might be also quite different. But how this is different, what dictates that difference? That's the question. And yep, so I hope this research has an implications to your research as well, community context or yeah. And also, the